wonderful introduction. And so I'm here today to present my thesis on parasites of the past, tracking changes in marine parasite abundance and diversity. So the central core of my thesis is how do we get reliable, accurate, and robust information about historical parasite populations for which we don't have contemporary data. And to start, I'd like you to view this. So if you were to go out in the Sound today, go diving, you might see a rock looking like this. It's pretty bare, some algae, some little tunicates there, and it'd be all right rock. But if you had gone to this rock 6 years ago, you would have seen something more like this, covered in pycnopodia, sea stars. And this difference, and this is the exact same rock. So where did the sea stars go? Did they walk away? Did they just not like that rock that day? Well, the truth is, in the course of 3 weeks from this photo to this photo, sea star wasting disease.
Turn your mic off.
I don't know where the mic is.
Causes
I am at the bottom of the
epidermis.
audio
Legs
video.
will literally walk away from the rest of the body and often leads to mortality. Sea star wasting is contributed to a viral disease. And some populations like Pycnopodia, there were severe and catastrophic losses. But it just didn't affect Pycnopodia, it infected the entire fields of sea stars across the west coast of the United States, Canada, into Japan. And if you were to put this occurrence, the sea star wasting disease, when it first hit on a timeline, it's something that looks like this. This little blue dot over here is the mass mortality event related to sea star wasting disease. And we have time on our x-axis.
What's Papa's cell phone number?
1900. And sea star wasting, it's at 24. So in a moment, I'm going to bring up a bunch
We'll
of
call his cell phone
dots.
so you guys can put it on speaker.
Each of those dots is a mass mortality event related to disease in some marine organisms. It could be mammals, fish, or invertebrates. And when you look at it, when you bring them up, it looks like this. These are all mass mortality events related to disease, extracted, reviewed by Harbell et al. In 2002, Merge et al. In 2013. And if you look at it, there seems to be a trend. If you look at 1975 and later, there's a lot of pink dots. That's a lot of mass mortality events related to disease. If you look before then, there aren't that many. So this pattern has led many disease outbreaks and parasite ecology. Marine diseases are on the rise. But we first have to take a step back and ask ourselves, are marine diseases on the rise? Is this pattern in actual disease, or is it just because we're looking now in an artifact of people?
Can you hear it?
Better tools to look at and find disease. And so we can just say things that happen now are disease when these were happening in the past.
Can I help?
So here's my outline for this. We will start with the present. What tools do we currently have in our toolbox? And all to answer the question, are marine diseases on the rise? And I will go into my first chapter, the past, part 1, using meta-analytic analysis to assess change in anisakid nematode abundance over time and see if we see some change. Then we will move into the past, part 2, authoring natural history collections to access data on parasites of the past and see and be able to look at more change over time. And then I will conclude with where do we go from here?
This means
And
yes, this means no.
we will take questions after I conclude the talk, and then I will give it thank yous after that.
Question.
Prepared for that. So to start with, what tools are currently? So the central core of what I've been interested in the past 2 years is, are marine diseases on the rise? And to answer this question, you have to ask yourself, do data exist to test this? We already know if they're on the rise. And if there isn't data, can we generate new data? Is it possible to go and find something that allows us to look back into the past? And if we create this new data, is it reliable? Will we know? Is this a true representation of patterns over time? So when we think about data, it can occur on two different scales. So on the y-axis, we have our spatial scale from local, like going right up here to Puget Sound and just looking there, to global, looking across the entire world. And we have a temporal scale, so like current, local, current events, 0 years ago, to back centuries. And in this space of data, you can think, and this data can cover, we use these scales to think about are marine diseases on the rise. And if you wanted to look, your first spot you would look for would be long-term data sets. This is the see it yourself type of data. The one where you go out and sample in the sound or wherever you are, and just look year after year for disease and parasites. And this would be like the gold standard of looking for change over time. And with long-term data sets, I'm going to show you an example. So here's an example from looking at an intestinal nematode of roach. And here we have prevalence, which is the proportion of fish hosts that were infected with the nematode. And on the bottom, we have abundance, which is how many were they infected with on average. So covering from 1970 to 2001. So if you look, there's not a super strong trend over time, but there's maybe been a more recent outbreak, and maybe these outbreaks will continue into the future from this set. But when I was looking at this and other long-term data sets, I had this question. What constitutes a long-term study? What is a long-term data set? So to start, I'm gonna ask you guys, what is your guess for the minimum duration of a long-term study? And just call it out. So for the minimum, we're going into a few decades. We're looking at a few decades. So then I want to know, what do you think is the average duration for a long-term study? So your ideals for what you would want as the minimum doesn't match what you would think is reality. So I had this question, and so I did a little back of the envelope meta-analysis where I searched in Web of Science for long-term and parasite in fish to get at what do fish parasite ecologists think as long-term. So I just extracted for papers that say long-term in their title or abstract, how long was the study? And I found the minimum to be 3 years. But the average was about 12 and a half years with about 9.5 years for error. So I think the max was around 30 to 60 years, so not bad, but when you put this in the scale of what kinds of data we can get, going out and doing it yourself, creating your long-term data sets gets you that corner. So temporally, you can't do it. They don't last long, but and spatially, you aren't going, due to funding constraints and other, like just people constraints, you aren't going to be able to easily create global data sets yourself for when you have to dissect fish. But people have been going out and looking at fish and parasites for a long time. And so while a single study might only last 10 years, that study might be from 1940 to 1950 or 2000 to 2010. And you can take all these studies and reanalyze them together in a method called meta-analysis. So this is the quantitative analysis of already published results, where you're pooling the results of numerous studies on a single topic or question and using proper statistical technique. So with this method, Ward and Lafferty, which was in 2004 and later updated by Chelsea, looked at the rates of disease literature over the entire literature for a taxon. So on our Y, we have the proportion of literature reporting disease. So it's how many papers were about disease over all the papers in a particular taxon. In this case, I'm showing the example of fish. And on our X, we have year. And so what they report is a Spearman's row, which is basically the trend over time. So here from Ward and Lafferty, I showed the example of fish, which is important because this part with Wood et al. 2010 updates this due to a previously unknown error. So after this update, looking at across many taxa, you find that turtles, corals, mammals, urchins, and mollusks have been increasing in disease from 1970 to 2001. And grasses, fish, decapods, and sharks have not been increasing in disease. So from Ward and Lafferty and later Chelsea, you might think diseases are on the rise at this broad taxonomic scale for just disease, the literature between 1970 and 2001. But that's 1970 to 2001. Can you go further back? Are there temporal biases in meta-analysis? This was another question I had when thinking about meta-analysis. So I did another quick back of the envelope meta-analysis calculation. So I had searched A in Web of Science to hopefully bring back most, if not all, papers that are in Web of Science. Then just took the count of publications per year and plotted it over time to see, are we temporally biased? And when you do that, this is what you get. So on our Y, you have total number of publications returned for that year. On our X is what year it is. And as you can see, we might be temporally biased towards recent years. In fact, if you create 1970 as a cutoff, 98% of publications are 1970 or newer, and only 2% are from before 1970. So you're clearly biased towards recent years. And this might have some factors of just total numbers of publications are increasing as population goes up, but also the ability to index publications on the web. So see this little jump right here? That corresponds to the internet. So when you think about it, the internet has really increased our ability to access publications, but going back in time, they aren't often been digitized or are available. So we go back to our plot of scales of data. We have our empirical down in the little corner. With meta-analysis, you can get from local to global, depending if a region is well studied or if there's one question that spans the world. And you can get about 50 years. So this 50 years, is that half century enough? And to really answer that, you have to think about change and what has gone on in the past 50 years. So with Lotz et al., here's a plot of the averaged abundance of the economically and ecologically important species over time on our X. And so here's now, or 2006 for the paper, going back 3,000 years, and this is the average relative abundance. So 100 would be pristine, with 0 being extinct, and you get depleted the further you go down. So, and all the different colors just represent different bays and seas across the world. So 50 years, that falls right there. So you can see, on average, ecosystems were degraded to 50% of historical abundances. And that's not getting back. That's only a proportion of recent ecosystem change. And if you want to get more change further back in time, you have to look. You might want to look at something else. So how do we go further back? Well, scientists have been asking this question for a while, and it spawned a field in recent decades called historical ecology, where they can look at artifacts of the past to infer historical ecological states, abundances, or other parameters. So one popular artifact of the past might be photos. Here's a photo from 1910 of a mola mola, or ocean sunfish. It weighs 3,500 pounds. And you can see that that is an impressive fish. And this type of stuff, these photos, and this information in historical ecology field was really brought to us a little over a year ago when Laura McClanahan visited for a seminar, where she displayed her amazing work on tracking changes in historical fish assemblages using photographs. restaurant menus, and looking at historic accounts from journals. And you can get historical information also from using genetic material, fisheries landing data, and really whatever you can imagine. If you can imagine it and you can find the resources, you can get some historical data. But I like parasites. And people don't take pictures of parasites unless you're in our lab or you're really grossed out by them and you want to know what that is. So there aren't many historical photographs of parasites. I don't believe there's any one parasite that would be 3,500 pounds in hanging from on a dot. Because in reality, parasites are about that size or smaller in often cases. They're small, hidden, hard to find, require dissection of fish to find most of them, and require some specialized skills and aren't often of general interest like fishing is and taking photos of fish. But what people do like to do is put fish in jars. So here's an image from right across the lawn in our fish collection, the Burt, and it has been estimated that there are over 8 million fish in jars around the world, and that's just from the 8 million number is just from 4 different institutions. And an institution is something like this, a large collection of fish. There's usually a public component and a hidden basement component where most of the collection is. And each of these institutions hold fish in jars, liquid preserved fish. And with this, people have been putting fish in jars for centuries. You can get some fish from the 1700s, 1800s, 1900s. You can find extinct fish, some fish, and all sorts of fish. And in each side of these fish, you have an ecosystem because parasites live in fish and they aren't just going to magically disappear, hopefully, when you put them in the jar. And so if you think about it, if you can extract the parasites from these fish that are in jars around the world dating back centuries, you can create a data set that looks like this. And you'd want to use all 3 of these tools to get at how are marine parasites and the diseases they cause on the rise in recent years. So we have our tools of long-term studies, analyzing published reports, and using natural history collections to cover scales that go from local to global, from now to centuries in the past. So that was the present. What tools do we currently have? And now I'm gonna move on to my first chapter, using meta-analysis to assess change in anesthetic nematode abundance time, and use one of these tools that we currently have to look at change over time. And then I will move into my second chapter, and how do we get at that large space for natural history collections, and then wrap it up with where do we go from here. So it's all framed around this question, are marine diseases on the rise? So for my first chapter, and here we start with chapter 1. So my first chapter is It's a Wormy World. Meta-analysis reveals long-term change in the abundance of anisakid nematode parasites of marine fishes and invertebrates. So while this is my thesis defense, I would like to take a moment and thank all the co-authors on this project for their support with it, along with Washington Sea Grant for their funding. So if you take a trip just a few miles down the road, you'll find Pike Place Market. And here you'll find fish on ice and flying through the air. And like I said, fish are ecosystems. So what's on that ice and flying through the air isn't just going to be a fish, but also any parasites in that fish. So here's an example of an anisakid nematode and a piece of salmon from the internet. So they were at a store, and that is a living nematode. And this isn't just some internet fad that a couple people have found. You can go and find them for yourself, such as Katrin and I did a few weeks ago in a store. So that dark little squiggle there is an anisakid nematode. And these anisakid nematodes are cool little critters. So they start their life being pooped out of marine mammal feces. It is eggs where they hatch. They're then consumed by crustacean hosts, which is then consumed by fish, and then consumed by the marine mammal, thus where they grow to be adults and complete the life cycle. You can see this transfer of the anisakid goes around, but people can enter this life cycle. When you consume a raw or undercooked fish, and that contains a living nematode, you can get a condition known as anisakiasis. So you will get symptoms that are like abdominal pain, nausea, diarrhea, vomiting, and might think you have food poisoning. And you might think, oh no, I don't want raw fish right now, but there are health standards to make sure that most fish is protected, that you are safe. So all fish that is served in restaurants and grocery stores should have been frozen for the proper amount of time to kill the worms, the anisakid nematodes, and thus making it safer to eat. So these anisakid nematodes, they're in the family Anisakidae. There are two main genera, Anisakis species and Pseudoterranova species. They show similar morphology and ecology. They follow the same general life cycle. And one of the big differences is their definitive host, that marine mammal they like to inhabit as an adult and reproduce. So for Anisakis species, they prefer cetaceans like dolphins and whales. And for Pseudoterranova species, they prefer seals, also known as pinnipeds. And this color scheme of blue for Anisakis and red for Pseudoterranova will show up again, but I've also made sure that the words are there so you don't have to worry about remembering which color is which. So my central question of my thesis is, are marine parasites and diseases on the rise? And with the ecologically and economic and human component of the anisakids, we have the question, is the abundance of anisakid nematodes changing over time? And specifically in the fishes and invertebrates that we, as people, can get infected from. And so to do this, we employed the meta-analytic technique, that tool in our toolbox. So to do so, we went to Web of Science, search for anisak or anisak to get Anisakis species worms. Herring worm is a common name of Anisakis. Pseudoterranova, which is also known as whaleworm, and Phocanema, which is an old name for Pseudoterranova. We conducted the search in Web of Science in October of about 2017. And this search resulted in 2,284 papers. And we wanted to take this 2,284 papers and get it to a workable data set with information that we could use to answer our question, is abundance changing over time? So you can do this in a couple ways. First, read 2,284 papers. If we had done that, you might not be sitting here today. You might still be reading some papers and being annoyed when you get papers that have nothing to do with what we actually want to answer. So instead, to get to our final data set that had all the information that we could get, we employed a winnowing process. So first, we screened the titles and we're looking for words such that were like about the wrong host. So like they were looking for worms and birds or people and not the fishes and invertebrates, or they were just looking at the wrong parasite, like a closely related parasite that's related to Anisakidae, or they just happen to mention Anisakis, Anisakidae somewhere in their paper. And so like sometimes we get a paper about Contrasecum, which wasn't of our two genera of interest. So after this title screening, which we did in at least duplicate with two authors looking at every title to make sure we aren't being biased by one author, by one person viewing these titles and throwing some out, we resulted in 1,336 papers, which is still a decent number of papers. But scientific literature has one more thing we can look at before screening the full text, and that's the abstract. So we then read the abstracts, looking for words about like experimental papers where they force-fed fish worms to see how many times worms can transfer from fish to fish. Or they were still, again, in the wrong hosts. Like maybe the title was just anisakiasis. You're like, I guess that would work for us, but don't know what the rest of the paper is about other than anisakiasis. And then you read it and it's about people. So we can still exclude wrong hosts. And after this process, we were left with 576 papers, with which we then read the full text, and we're looking to make sure it had sufficient information to include in our final data set, which we were looking for a specific location, let's say the Puget Sound. Ideally, they'd give a lat or long or an identifiable location where we can extract a latitude and longitude to get a specific location, what fish host they were looking in. So like salmon, and specifically like Chinook salmon, not just salmon in general, when they collected those fish. So basically, what year were the fish collected, so we can put it on our timeline because we want to change over time. And if the fish were just collected and we don't know when, we can't accurately estimate when they were collected because sometimes science takes time and the year of publication is not necessarily the year the work occurred. We then needed an estimate. The paper needs to report the host size because size is a known correlate to parasite abundance. And if we can't control for size, our results could just be changes in fish size over time of what hosts and fish we have data from. We need to know what parasite they actually found. So was it an Anisakis species or a Pseudoterranova species? And we'd accept from genera down. How many parasites there were. So we needed an estimate of the abundance or how many worms on average per fish, or some information to calculate that, such as the proportion of hosts infected, and of those infected hosts, how many worms they had, because we can multiply the two and get an estimate of abundance. And we also need to know the error associated with that. And the fish had to be unprocessed. You couldn't get a fillet from the market and look at that, because that might have some screening and humans removing fish as they fillet fish. So after this process, we had 123 papers with usable data. And of these 123 papers, they could contain multiple abundances for the worms in different hosts or locations or years. So you can get, so when you split this 123 into which contain information on Anisakis and Pseudoterranova, the sum of these two numbers is not 123, but more, because you can have overlap. So 112 of the papers had information about Anisakis species abundances, and 39 had information about Pseudoterranova species. And from these papers, we extracted what are data points for us, which was the parasite abundance for every unique combination of host species, parasite species, location, and year we could from that study, from those studies. And this resulted in 526 data points for Anisakis species and 229 data points for Pseudoterranova species. And we use these data points, these estimates of abundance, to create a meta regression model where we modeled the fourth root of abundance, which we transformed to meet normality assumptions of the model. And we modeled against host length, because we know size of a fish influences parasite abundance. The year that the hosts were collected, to answer our question, is abundance changing over time? But we also controlled for different demographic factors of the fish, let's say. So what species of fish was being looked at, which portion of the fish was being looked at, was it the entire fish, was it just the muscle or the viscera, where in the world the fish was, were you in which we use as FAO regions, so were you in North Atlantic, South Pacific, Japan, off the coast of Japan. How you look for your parasites. Did you use standard technique where you squash muscles and organs? Did you digest your fish in acid and pepsin to just leave the nematodes behind? Or use some squash and UV light, because nematodes often fluoresce under UV light. We also controlled for which paper did this data point come from. So when you look at these plots from this study, from these models, we have abundance on our Y, so the number of worms, and this has been length standardized so that the trends are, all the data points are comparable. And we have on our X year, so we have and host species is, and the different colors are host species, which between the two genera, there were over 200 different potential host species. So listing out each individual host species, you can't tell the difference in most of the colors. So just know their different colors are different hosts. And the different shapes, whether it's circle, triangle, square, or plus, represents different portions of the body that they looked at. So for Anisakis species, abundance increased 208-fold. Between 1962 and 2015, there's been a large increase in abundance. And to put that in perspective, here's a fish with a worm. That little green dot that you probably can't see is a little encysted nematode in this fish. And if you were to scale that up to be 208-fold times larger, you would get a fish that looks with a worm that looks like that. So that's been a major increase over the past half century. But when you look at Pseudoterranova, abundance didn't change. There was a constant abundance over time. We did not detect a significant change. So our study was merely correlational. We were looking for trends over time. We didn't relate it back to any explanatory variables, but we have created some hypotheses as to why we might observe these changes. So why? If you think about it, anisakid nematodes follow this complex life cycle. And with that, you can have a Y enter at some point in this life cycle. So first you might think marine mammals. They might have changed in recent years, and in fact they have. In 1972, the Marine Mammal Protection Act was implemented in the United States, protecting marine mammals. And then in 1982, there has been an international moratorium on whaling. And this has allowed for large increases in both whales and pinnipeds, which, if you think, these whales and pinnipeds are the life cycle bottleneck of anisakid nematodes, you would expect these increases to have corresponding increases in the fish. So if we plot out where the Marine Mammal Protection Act and International Moratorium occur on our data frame, on our data set, they occur about here. So it could be our data strongly overlaps with the time of protection, but we have a lack of data before marine mammal protection. Remember, Pseudoterranova didn't change. So even though there's these protections, and in fact, sometimes stronger increases in pinnipeds compared to cetaceans, abundance of Pseudoterranova didn't change. So maybe pinnipeds just aren't the rate limiting step in the Pseudoterranova life cycle, while cetaceans are for Anisakis. Or maybe there's something else going on. Maybe there's multiple changes, and it's a combination of many factors. But if we move along in the life cycle, our next host, or the first host after you're hatched from an egg, is our small marine crustacean. So over the past 50 years, there's been changes in temperature and productivity that can change abundances and distributions of different crustacean hosts. And maybe these can lead to changes across the life cycle of anisakid nematodes. And maybe there's differences between which crustacean hosts Anisakis and Pseudoterranova uses to result in these differing patterns. But then if we move along, we'll reach the fish. Maybe people have done something to change how fish acquire infection. So maybe pollution or changes in temperature have altered immune responses of fish, or the ability of nematodes to insist or re-insist in fish. And we can keep creating hypotheses as to why, but at the end of the day, we can't answer why with our study, but we can, our result of a 208-fold increase in Anisakis species nematode abundance has very important implications for human health. So increasing abundance of Anisakis nematodes can increase the potential risk of there being a worm in your sushi. And then your risk for anisakiasis might go up with this increase in abundance. But even if you don't eat fish, there are things that like to eat fish, such as the marine mammals. So with increasing abundance in the fish and invertebrates, marine mammals can acquire greater burdens even if they're eating the same amount of fish, but if those fish have more worms, they can now acquire more worms. So this is an image from a necropsy of the stomach of a stranded sperm whale, and all those worms are anisakid nematodes. And even if you aren't a marine mammal lover, you might worry about people in fisheries profitability. So increases in anisakid nematode abundance you might end up with fish looking like this. So that's almost more worm than fish if you look at surface area image from right here. And in fact, and even me, who would love to buy fish if it's affordable with worms, uh, wouldn't particularly want to buy a fish like that unless it was free, and then just bring it back to lab and be like, look. And everyone will know if I ever find one that looks like that. And if you think, and with fisheries profitability, Bao et al. Did a study in Spain where they asked consumers and found that, where they did a questionnaire for consumers and found that consumers in Spain are willing to pay more for certified anisakis-free fish. So with increasing abundances of anisakis, the ability to have anisakis-free fish declines. And fishermen can lose profits if they could advertise that their fish are parasite and anisakis worm free. And with that, let's go back to my central question. Are marine parasites and the diseases they cause on the rise? Well, I have two examples to add to the literature. One of anisakis species having a marked increase over recent years, and Pseudoterra nova largely staying constant. And so while I can't make a broad scale sweep saying, yes, because there's one that didn't change and one that goes up, so on average it's going up. Because that's just two examples in the case of many, many thousands, tens of thousands, over hundreds of thousands of marine parasites. So we still have the question to answer, are marine diseases on the rise? And that concludes chapter 1, using meta-analysis to assess change in anisakis nematode abundance over time. So now we're going to move into part 2 and look at extending our data sources to the past couple of centuries by unlocking natural history collections. And then I'll finish up with the future. How do we bring it all together? Where are we going from here? So, unlocking natural history collections to access data on parasites of the past. So if you remember, or see here, natural history collections represent this wide space of data from local to global scales, from now to centuries in the past. And with over 8 million fish in jars around the world, this is an impressive potential source of data. But before we access these data, we have to ask ourselves, can we use these fish in jars to learn about parasites in the past? So what is this fish in a jar? So they go through this process called fixation. And here's how you do it in 4 simple steps. First, you place it in a solution of 10% buffered formalin for a period of time that varies depending on the size of your fish. After it is properly fixed, you will place it in a tap water rinse for 24 hours. Then you'll repeat that rinse for another 24 hours. And finally, place that fish in 70% ethanol in a jar if it's a smaller size fish. If it's a larger fish, you might grade the ethanol up from, say, 30, 50, 70 to prevent dilution and make sure your fish is preserved over time. So after this process, you're left with a fish that looks like this. So here, fish in a jar, and let's imagine we could just zap away all the fish tissue and be left with what's left that's not fish, the ecosystem inside, the parasites. And looking at this, these parasites can give you the general shape of the fish, where different things are in the fish. If you know, like adult nematodes like to be in the intestine and stomach, and there's a bunch of nematodes right there, adult nematodes right there. That might be your stomach and intestine outline. Or monogenes in the gills, a little gill archy thing looking right there. And these parasites represent many different taxa. So you've got some flatworms, so like monogenes, trematodes, the flukes, cestodes, the tapeworms. You have nematodes, you have acanthocephalans, or the thorny-headed worms, and you have crustacean parasites such as copepods and isopods. And there's this large diversity of parasites. And so a fish leaves behind its ecosystem, and you want to know if preservation affects it. Well, we want to know it for natural history collections, but all parasite ecologists would want to know about collecting fish in the field. Let's say it's a remote place like our lab does on Christmas Island. And how do you get these fish dissected? Do you do it in the field fresh after collection, or are there ways you can bring these fish back? So the Czech Republic, Kovač et al. Had this question, and they looked at the effect of fish preservation methods on field preservation methods on detectability of parasites. So they employed a simple experimental approach where they collected a bunch of fish and then split those fish into a few different treatments. They either kept them alive, froze these fish, put them in ethanol, or preserved them in formalin. And what they found was that preservation can decrease detectability. And for copepods and acanthocephalans, there was no change. And for the trematodes and cestodes, the tapeworms and flukes, they found decreases in detectabilities. But they didn't have enough information to confidently say about nematodes. But if you think about it, they were looking for field preservation methods. And those, because they don't want to have to dissect fish in the field where it might be hard or difficult or expensive, time consuming. And if you think, the fish in the natural history collection is preserved in a way that is known to best preserve the fish tissues. And with most fish in collections that are preserved in this formalin and stored in ethanol, because it's the best way to preserve fish tissues, we want to know, is this also the best way to preserve parasite tissue? Do the parasites also remain better if you do the best method for preservation? So this brings me to my second chapter. Liquid preservation causes minimal loss of parasite detectability in fish specimens, a new approach for reconstructing parasite communities of the past. And again, I'd like to thank my co-authors for their help conceiving this experiment and providing guidance, helping dissect fish, and also the Alfred P. Sloan Foundation and UW Innovation Imperative for funding support for this project. So we, like Kovač et al., took a simple experimental approach. So we went out, found got some fish that represented varying different sizes. We visually paired these fish according to size. And for each pair, we flipped a coin to determine which fish went into which treatment, which fish went into one of two treatments. So split into two treatments, and then asked. Then with one treatment, we just simply dissected the fish from the state they were in. In the second treatment, we preserved the fish first and then dissected according to our parasitological dissection methods. So we did this for 3 different species of fish: English sole, which we collected out here in the Sound with the fisheries ecology class in 2018. And then we also used Eulicon and Elastopolyp, which were provided by the UW Fish Collection. So we wanted to ask our question, and that was, and look for differences between these treatments. And with this, you might expect that we might want to be finding a null result. And if you want a null result, you might need to make sure you have sufficient power to be able to detect the null result. And so that your results, you're confident that there's no difference and you just were an underpowered study. So we needed to ask, how many fish per species did we need to dissect? So we ran a power analysis where we simulated based on simulations. So we generated data using different starting means, which range from 0.5 to 5 in 0.5 increments with then simulate different differences in abundance between treatments from 0 to 2.25 and 0.25 increments, and then across different sample sizes, with sample size of 50 over the entire 50 fish total per species, 100 fish, 200, 500, or 1,000 fish total. So those numbers would be divided into for per treatment samples. Then we ran our generalized linear model to detect differences in abundance. We repeated this 500,000 times. So for every combination of starting mean, difference, and sample size, we ran 1,000 simulations. And you recorded if that our analysis of that simulated data was showed a significant result and recorded that to create our power, which is the proportion of times if with that given starting mean, difference, and sample size, so different effect sizes, what proportion of time would you expect if those were your underlying conditions, you would detect a significant result. So after doing that, we ended up having a plot like this. So on our y is power, which is the proportion of times that the test would be significant. The 0.8 is a general, is a rule of thumb cutoff, as you'd want at least a 0.8 power for your analysis. On our x, we have effect size. We're measured in Cohen's d, which is the standardized difference between means accounting for a pooled variance. And Cohen, in his paper in the 60s, recommended 3 general cutoffs: a 0.2 for a small effect size, 0.5 for medium, and 0.8 for a large effect size. And then, because we want to know how many fish to dissect, we have our different sample sizes that we simulated, ranging from 50 in this light yellow to 1,000 in the dark green. And we found that to detect a small effect size, we would need to dissect about 1,000 fish per species, or 3,000 fish over the entire study. To detect a medium, we would need to dissect about 100 fish per species, and to detect, if we only were interested in a large effect, we would need to only dissect 50 fish per species. So smaller effect size, being able to detect smaller effect size is ideal, but there's a trade-off between smaller effect size and sample size. So to get a small effect, to be able to detect small effect sizes, I would need to dissect 3,000 fish. And if we were doing that, see you in 6 years, something like that. So we took the trade-off and went with 100 fish per species so that I could finish today and still be able to detect moderate effect sizes. But I've been talking about small, medium, moderate. What does that actually mean? So a small effect size is a difference in abundance of 0.5. So half a worm difference on average between the 2 treatments. For a medium effect size, you'd have a 1 1/4 worm difference. And for a large, you'd have 2 worm difference. So our analysis was our planned experiment would allow us to detect a difference of between greater than 1.25 worms on average. So knowing this, we went out and dissected fish, which looks something like this. You start by making a ventral incision from the anus to the gill isthmus. You open it up and remove all the internal organs, place it in a dish. You also remove the right side of the gills. And then depending on how many fish were in a bag when I thawed it or preserved the fish, you would end up with some trays looking like this with the internal organs, the bodies, and the gills. Then you would start going through all these. So for the internal organs, you do organ squashes, where you place the organ between 2 glass plates and just squash it so it's thin enough to easily pass light through. And here is some liver from, I think, a pollock. And if you notice, those are 2 anisakid nematodes staring you right in the eyes. And then, if you look then for the intestines and stomachs, you open those up and look through the contents through a Bogaroff tray and extract all parasites you can find. You'd use a similar process for the gills, all on dissecting scopes. And then for the bodies and fins, we use the process called candling, which is just passing a strong light through the body of the fish to look for parasites, such as these Clavonema right here. So traditionally, parasitological methods have you fillet and skin at least one side of the fish and squash it to look for parasites. But we wanted to use this technique on museum specimens, and taking literally half a fish away is not ideal for the longevity of collections. Thus, while this method is a little less, we don't have as much ability to detect parasites using this method, it's safer for the collections, and thus we had to make the trade-off of keeping the collections healthy into the future and using a less than ideal method. So across all of this, we found that of the parasite taxa that we were able to run, that we had sufficient abundances and prevalences to run our models on, showed no significant differences in detectability between the two treatments. And we can now look at the individual parasite taxa that I found. So this plot is of the standardized regression coefficient on the X. So basically, it's a standard effect size. So anything outside of the dotted lines is significant after Bonferroni correction, and anything inside, any of these points inside, are non-significant. On our Y, we have our different parasite taxa that we detected, and you don't really have to worry about the numbers. Just know that if it's over here, we had increased detectability in the preservation treatment. And if a dot lies over here, we had increased ability to detect the parasite in the control treatment or frozen fish. So for English sole, we found that no differences for trematodes, largely no differences for most of the nematodes, and no differences for a leech. And the one time we did find a difference was for a cuculanus species, cuculanus nematode, which is an adult nematode infecting the sole's stomach and intestine. And we had an easier time finding it in the preserved treatment. For pollock, we found largely no differences for the trematodes, no differences for the nematodes, no differences for the cestodes or tapeworms, and no differences for the acanthocephalans or thorny-headed, spiny-headed worms. And the one time we did find a difference was for an adult trematode that inhabited the stomach and intestine of pollock. And we found it easier to detect it. There was increased detectability in the control treatment. For Eulicon, we found no differences in the trematodes, largely no differences in the nematodes, and no difference in the cestodes. And we found increased detectability in Pseudoterra nova species, which is a larval nematode that also showed up in my meta-analysis, if you remember. So, looking at this, we have individual parasite taxon level estimates of differences of how preservation affects their detectability. But we wanted to know what happens if you pool across the parasite taxa. Are there general trends across taxa that elucidate themselves? So we wanted to use meta-analytic techniques and basically analyze my own results to look for tendencies in parasite detectability across all parasite taxa, which life stage that parasite was it a juvenile or an adult? Because juveniles are often encysted in the host tissue, which can create some protection from the effects of preservation. What group of parasite you are. So were you a nematode, a leech, a fluke, a tapeworm, or an acanthocephalan, or was it specific by life stage and parasite group? So this method of analyzing your own results is not common. It's been pioneered by Chelsea in her PhD work. And so I borrowed on those techniques here. So overall, on our X, we have which parasite group you are. We have the effect size on our Y. So with positive effect sizes being increased detectability and preservation, negative being increased detectability and control. And the different colors represent, is it the combined just for the group or all parasites? Is it the adult parasites or the larval parasites? And each bar is the mean and standard error. And so, are there differences across all parasites? No. Are there differences by just general life stage across all the parasite groups? No. In the trematodes? No. Nematodes? No. Leeches? No. Cestodes? Adults are easier to find in the preserved treatment. And in acanthocephalans? No. So largely, collections represent the preservation process has minimal effects on detectability at these larger scales. We have to remember, my study could only detect a medium effect size. So we can't say if there are small changes of less, let's say, of half a worm change in abundance between the two treatments. And so just keep that in mind as we move into our caveats. What can my study can and can't say? So first off, I've found no monogenes or copepods in either at all or in sufficient number to run my models. So we can't really say what's going on here with them. We can create hypotheses such as monogenes being small, soft-bodied, often on the fins or in the gills, are likely could show decreased detectability in preserved treatment due to just being the harshness of the formalin and ethanol and dehydration because of them, or just being knocked off as you handle the fish as you go through the preservation process. And for copepods, they're protected by a hard, and other crustacean parasites are protected by a hard exoskeleton. So the formalin and ethanol and tap water rinse might not be particularly detrimental to them, but people are handling these fish, and these parasites are often can be knocked off of these fish, so they might show decreased detectability just because they've been lost. And if you think about people run these collections, so they can get a fish in that looks something like this. So all these dark blotches is a Clavinema worm, a parasitic worm. And when you think about it, a collection manager or a collection in general can choose to preserve or not preserve this fish, depending on how that collection is run. Some collections might want pristine examples of the fish to be held in their collection, and limitation of space might mean they can't take all the fish, so they have to be picky. And they might exclude obviously heavily parasitized fish and bias collections to be lower in parasite abundance. Or maybe you're a collection that can add some freedom and this looks super awesome. So you preferentially preserve these fish because it's different. It's not the standard, and you might not recognize that these are worms, and you might wonder why. So you preserve it to later ask someone else. Or maybe you're a special type of collection, such as we have over here across the lawn at the Burke. So the Burke is one of my favorite places to hang out, down there in the basement. There's tons of fishes in jars. In fact, there are over 12 million specimens between the larval, adult, and otolith collection. It is the largest collection in North America, mainly focused on North Pacific and Alaskan fishes. And many of these specimens come from scientific sampling. The Alaskan Fisheries Science Center of NOAA deposit, the Burke is their repository for their fish that they collect in their trawls. And with scientific sampling, you're just collecting fish, and you organize it by species and by tow. And then when you preserve those fish, they just all get preserved along, often all get preserved along. So what you have in the Burke are more scientifically representative random samples of fish populations. But there's one more caveat we have to think about. Years of storage. So I stored my fish in ethanol for about a week because while the fish on shelves in the collections can be there for years, decades, a century or more. And well, given the limits of my lifespan and the degree and wanting to finish and not just sit waiting for a fish to be a year old on a shelf, in my study, did not look at how the different years of storage affected that. But our lab with these crack team of people is looking at, does parasite detectability change over a decade of ethanol storage? We went out, collected fish, English sole from the Sound, and put them in jars, and about every 2 years, we will crack open one of those jars and dissect the fish so that by 2029, we'll have a decade timeline and can answer what does a decade's worth of storage do to parasite detectability. And with that, we can come back to this plot. I have not in this chapter answered are parasites and the marine parasites and the diseases they cause on the rise, but I have opened up a large space to ask this question, to look across ecosystems, species, And in general, how do these, and to be able to use these natural history collections and build off of this from here. So that was my second chapter. And now I'm going to bring it all together, and where do we go from here? And looking in how this all relates back to our marine diseases and the parasites that cause them on the rise. So we have tools to look back in time and truly answer, is marine disease on the rise? So are marine diseases on the rise? Keep it posted. So my thesis found one increase and no change in other taxon, and then opened up the tools to ask this question across many host and parasite species. But you can't answer this question without looking at specific changes in specific locations across the world, can then scale up to global over the past century or more. But we do have the tools to now look at multiple lines of evidence. This, in historical ecology, the ideal practice is you use one method to estimate change or look at a question or historical parameters. And then, in case that source of information or data is biased, you would use a complementary but independent source of data to make sure your patterns are consistent. And so I have a meta-analysis chapter for anisakids. And we now have the tools to look at natural history collections. So my lab mate, Natalie Mastick, is doing just this. She's pulling out anisakids from museum fish from right here in the Burke to look at changes over time in anisakid nematodes. And we can then collaborate her results and my chapter thesis results and see, are the patterns consistent? Are there potential local scale differences in change over time revealed from Puget Sound fishes that might not be captured in my global data set? And her chapter is part of a larger project that we call the Parasites of the Past project, or the POP project. So this is collaboration between the Wood and Tornabene labs to look at a century of change in Puget Sound. We are dissecting fish from 10 different species, all that span across the food web, that are all located right over there in the Burke. And so we can look at change in Puget Sound, local, what have parasite populations been doing right here. And with this, we can go back to the Burke and start thinking not just about the Burke, but natural history collections in general, and our projects that we're been building up on from my chapters, and start thinking of questions like, what drives parasite change? Is there change? Have we lost species diversity, gained parasite species diversity? Can we detect extinctions of a parasite? Maybe we'll find a historically extinct parasite that hasn't been described. New species of parasite that we just don't know. And we have the tools to look in these fish in jars and enumerate their ecosystems and really start thinking more about our marine parasites and the diseases they cause on the rise. With that, I'll leave you with a quote by William Wordsworth. Life is divided into three terms: that which was, which is, and which will be. Let us learn from the past to profit by the present, and from the present to live better in the future. Thank you. Yes. So we focus on wild fisheries only because aquacultured fishes are often fed prepared foods. So we did return some aquacultured fish papers, but they're largely free of parasites because what they consume, because it's a prepared feed, often doesn't contain the living anisakids from like the crustacean hosts that they can acquire. So often there's no anisakids in aquacultured food, or very minimal low abundances from just a random, let's say, krill or shrimp getting into a cage and then a fish eating it. So all of my trends are for wild fisheries. Martini. So we looked for if there was a host-specific drive. So to do that, we excluded one by one each host from our model to see if our results changed, and found that there was no host that changed the patterns we found over time. So I didn't show this here, but our data is heavily biased towards the North Atlantic and Mediterranean, and we have less representation of data from, and we have a decent amount of data from the Antarctic, but we have less from the Pacific, Japan, and Australia, and Indo-Pacific. Chelsea.
So you focus mostly on parasites. Would you make the same predictions for change into the future for pathogens? Do you think that there's going to be a diverging trajectory for macroparasites?
I would say there's probably going to be differences. So the microparasites like bacteria and viruses likely are often going to be more host-to-host transmission unless this, well, you can have host-to-host and metazoan. There's a large number that are complex life cycles. So the host-to-host, the complex life cycle parasites are often more sensitive to environmental change because they have, let's for the anisakis, for example, 3 different things that could respond to the 3 different hosts that could respond to the environment: mammals, invertebrates, or fishes. And depending on that change, if there's a sensitive link in that chain, you can see maybe not an increase, but extirpation of that parasite because you cut out a link and that life cycle fails, and you see declines or loss of that parasite. Whereas the more directly transmitted, like microparasites and pathogens, might be less sensitive to that. They can exist, let's say, in the dead host tissue for post-mortem for a little bit and still be infectious. So you might, in the long scale of environmental change is extreme, where everything dies, everything's going to die. But in the meantime, they might be less sensitive to that, or maybe respond positively to more stress on the host. Any other questions before I move into my thank you video? So I was talked for the past hour, and as many of you know, I'm not a talkative person. So I chose at 10 o'clock last night, instead of making a short PowerPoint, to make a 3-minute video, which took a little longer than making the PowerPoint, but allows me to rest my voice before answering questions from Mark, Chelsea, and Luke. With that, let me find it.
Thank you all for coming. We're now going to do Evan's closed door session, and hopefully in an hour there will be celebration of backwards art successes. Thank you all online for joining us. We're going to cut out now and go to our closed door session, but we're really grateful that you were able to make it.